Right, so we're going to start in on the digestive system. Okay, we're going to start by talking about structure and function. So like normal, we're going to do all the anatomy you need to know in order to understand the function of the digestive system. And then we're going to follow food on its path from our mouth to the anus, all right? So we're going to start with chewing and swallowing. So we'll talk about the mouth, pharynx, and esophagus. Then we're going to talk about digestion and absorption. We'll talk about the stomach, then the small intestine, and then we'll take a little detour, right? And we're going to talk about the accessory digestive organs, the pancreas, liver, and gallbladder. And then we're going to talk about the large intestine. Right, so here we have a straightened out, simplified GI tract. So here's the mouth, okay, and then here's the anus. Your GI tract is an external body surface that runs from your mouth to your anus, right? And so what goes in your mouth is going to get chewed, okay, swallowed, and end up in your stomach. Okay, so the little brown chunks are your food, and this is where digestion starts. We do a little digestion in the mouth of carbohydrates, okay, but digestion really doesn't start until we hit the stomach, okay, and enzymes are what are doing the digestion, okay, and what macronutrient in particular gets digested in the stomach? Wait, I couldn't hear what you said. Protein, you're right, yeah. So protein gets digested in the stomach, okay? And then we're going to hit the small intestine, and this is where the majority of digestion and absorption occurs, okay? So here's the purple absorption. So this is moving those macronutrients from the external body surface into the internal body compartment. In order to absorb them, we have to make them small enough to cross those epithelial cells, okay? And those epithelial cells that line your small intestine are just like the epithelial cells that line your proximal tubule, right? So when we talked about absorption of things like glucose and amino acids from the kidney tubule, that's how they get absorbed from your GI tract as well. Okay, and again, digestive enzymes are what are going to break those large macromolecules into small enough monomers to get absorbed. Okay, and then whatever does not get digested and absorbed, okay, is going to be ejected as feces. Okay, you want to move food in the direction from your mouth to your anus. Okay, so one of the major roles of the GI tract is motility. Okay, so that's peristalsis. Not only does it move food through the GI tract, but also helps mix it. Okay, mix it with the digestive juices. Those digestive juices are secreted into the GI tract. So secretion is also a major role of your GI tract. Okay, and then of course digestion and absorption. So those are the four major roles of your GI tract: motility, secretion, digestion, and absorption. Although we probably should add up here protection as well. Because remember, we were talking about the immune system. Your gut-associated lymphoid tissue is the largest lymphatic organ in your body. Okay? You invest a lot of your immune system into protecting your body by screening what you put through your mouth and ends up in your GI tract. Hey. So motility is due to muscular contractions. And those muscular contractions both mix the food as well as move it on its way. Okay, so we've got propulsive movements and mixing movements. So the propulsive ones are pushing it forward through the GI tract. Mixing makes it with digestive juices to promote digestion, and also make sure you expose it to the GI wall so that it gets absorbed. Right, so you want to make sure you have plenty of mixing going on, as well as propulsion. And this is all done by smooth muscle contraction, except the upper esophagus. Okay, your upper esophagus is skeletal muscle. But then you hit smooth muscle, so it's not under your conscious control. Okay, so your GI tract right now is doing all sorts of mixing and propulsion actions that you don't consciously control. Okay, secretions from your GI tract consist of water, right, in order to dissolve food particles, electrolytes. Okay, so in the stomach, you release a lot of hydrochloric acid, so that's hydrogen ions and chloride ions, so those are electrolytes. And then in your small intestine, you release a bunch of bicarbonate. Okay, so you make your stomach contents really acidic, and then you bring the pH back up to neutral in the small intestine. Of course, you secrete a bunch of mucus in order to protect your GI tract from all those secretions, because you don't want to digest your GI tract. Okay, digestive enzymes get secreted, as well as antibodies. And what class of antibodies gets secreted in the GI tract? Who remembers? IgA, excellent. Okay, so IgA antibodies are the class of antibodies that cross epithelial barriers, like your GI tract. Okay, some of your secreted products get reabsorbed. Okay, your GI tract's really eco-friendly. So the amylase you secrete in your saliva, right, that you used in your experiment in lab, what happens to it in your stomach? What gets digested in the stomach? And what's amylase? Oh, protein, right? So it gets digested, and then you're going to absorb those amino acids, right? So your digestive enzymes get digested themselves, and then you reabsorb those amino acids. Okay. So digestion is the biochemical breakdown of complex macromolecules into absorbable units. Okay, so the enzymes in your GI tract do hydrolysis. And just as a reminder, since we haven't talked about this in a long time, here we've got a disaccharide, maltose. Okay, and by introducing water, we can break the covalent bonds that's holding the two glucose monomers together to form maltose. Okay, so our digestive enzyme maltase would bring about this chemical reaction, right, which is called hydrolysis, because we're breaking a covalent bond by introducing water. Okay. Our carbohydrates, so if you eat a lot of complex carbohydrates like you're supposed to, rather than just a whole bunch of glucose, you have to break down those complex carbohydrates into monosaccharides in order to be absorbed. Okay. Proteins have to be digested into di and tripeptides, as well as just amino acids. Okay, so just a simple amino acid, or two held together, or three held together. Okay, and then fats get broken down into glycerol and fatty acids in order to be absorbed, which is our fourth major role of the GI tract. Okay, so those small units from digestion, as well as with water that you've either drank, okay, exogenous water, or water that you've secreted into the GI tract, as well as any vitamins you've taken in and electrolytes you've taken in, are going to move from the lumen of the GI tract into the internal body compartment. Okay, so here we have the intestinal lumen, which is an external body surface, and it's lined by epithelial cells. And these epithelial cells are called enterocytes. Okay, and just like those proximal tubule cells, the apical membrane that faces the intestinal lumen has microvilli. And what does that tell you about their role? You see microvilli and epithelial cells, they are specialized for absorption. Okay, because they're increasing surface area for absorption to occur. And they are what separate the external body surface, the intestinal lumen, from the internal environment, which is your bloodstream or your lymphatic system. Okay. Carbohydrates get broken down to monosaccharides, proteins get broken down to amino acids, and then they're absorbed through those epithelial cells by both active and passive processes that we'll talk about. Okay, fat is a big problem. Okay, fat is hydrophobic, whereas all your digestive secretions and all your digestive enzymes are water soluble. Okay, so fat needs to be emulsified, and it gets emulsified by your bile salts. Okay, so you've got your big fat globules, bile salts emulsify them, and that breaks them up into small enough droplets that the digestive enzymes can work on them, right? And then they get absorbed, and then they get packaged inside the enterocytes. And then they get released by exocytosis, because you don't want to drop a whole bunch of fat into your bloodstream, because what will happen?
Yeah.
You die. Right, exactly, you die. It comes out of solution, okay? And it's just going to form bigger and bigger fat globules. So in this picture, these little green molecules are proteins. Okay, so you package dietary fat with protein into what are called chylomicrons. They're too big to get across by just regular facilitated diffusion or something like that. So they're released by exocytosis. And then remember we were talking about the lymphatic system? We said one of its roles is to transport absorbed dietary fat. Well, here's why. Okay, because those chylomicrons are too big to get into the capillaries. Okay, so they end up in the lacteals instead. And we'll talk in more detail about digestion, absorption, and fat. Just a preview. Okay, here we've got the organs of the digestive system. So we've got the mouth, and we've already talked about our mouth because we talked about our tongue and the sensory organ associated with it, so our taste buds. Okay. We've got salivary glands, which are accessory or associated structures. Our salivary glands produce saliva. Okay. And then we have our pharynx, which is our shared passageway for our respiratory tract, which is being shown in this light purple color, and our GI tract. Okay, so when we swallow, we cover our trachea so that the food and water goes down our esophagus. Okay, and then the esophagus empties into the stomach empties into the small intestine empties into the large intestine empties to the external environment, right? And then we've got our liver as a major accessory digestive organ, okay, our gallbladder and our pancreas being shown. Throughout the GI tract, that wall has a similar structure, and it has 4 major tissue layers: the mucosa, submucosa, muscularis externa, and serosa. So the mucosa is the innermost layer that's facing the lumen. Okay, and then the serosa is the outer layer. So let's look at a cross-section of the small intestine in particular, because we'll spend most of our time talking about the small intestine. That's where the vast majority of digestion and absorption occurs. Okay, so the mucosa is that layer that's facing the lumen, right? And then we've got the submucosa being shown in purple, and then we've got the muscularis externa, and then that outer serosa layer. Okay, so the muscularis layer has smooth muscle. Some of it's circular, some of it's longitudinal, and that allows for both mixing and propulsive movements. Okay, so having those 2 types of smooth muscle, some of it's circular, some of it's longitudinal, allow for that peristalsis, right? So the mixing and movement of food. Right? Notice we have lymph nodules associated with the mucosa. Those are to both pick up, absorb digestive fat, but also to be screening and scanning what you're putting in your mouth. Okay, you've got B cells in there. They're going to be releasing IgA antibodies, okay, because that's one of the secretory products. And then that mucosa layer, the portion that's facing the lumen directly, is lined by those enterocytes. Okay, and those are the epithelial cells that are specialized for absorption. Okay, the submucosa has the larger blood vessels. Right? The muscularis layer has the nervous tissue, which is part of the enteric nervous system, and then the serosa is mainly just connective tissue. Okay. Any questions about that structure and function? Pretty basic stuff. Okay. Yeah.
Should we know the order of layers there?
So yes, definitely know that the mucosa faces the internal body compartment. Yep. Right, so let's talk about our mouths. So here we've got the organs of chewing and swallowing. So we've got the tongue, we've got the teeth, which are all present in your mouth. We've got the pharynx, which is the shared passageway for both the GI tract as well as the respiratory tract. So what's being shown in this figure, being pointed out here, is also the epiglottis, okay, which is going to cover the trachea when you swallow. Also notice there's an upper esophageal sphincter, so that when you're breathing, that stays closed so that you don't get a bunch of air into your GI tract. Because if you get a whole bunch of air into your GI tract, how does it come out? As gas, okay? You're either going to burp or you're going to have flatulence. So people who chew a lot of gum end up swallowing a bunch of air, right? And tend to be gassier than people who don't chew gum. Okay, so your mouth, you've got your lips that form the opening and help procure, guide, and contain food in your mouth. So if you're talking while you're eating, they don't contain the food very well, okay? And then you end up spraying whoever you're talking to with food. Okay, your lips are obviously also important in speech. And remember, they had really high acuity, right? They had really small 2-point threshold discrimination in the sensory system lab. So they had this really well-developed tactile sensation. Okay, your palate forms the roof of the oral cavity, which separates your mouth from your nasal passages. Okay, and then your uvula seals off your nasal passages when you swallow, so that food doesn't end up in your nose. But if you laugh, oftentimes when you swallow, sometimes food will come out your nose. Right? And there's always that like 3rd grade boy who can get spaghetti to go up and string it through the nasal passage. Okay, the tongue forms the floor of the oral cavity. It's made up of skeletal muscle, so you can consciously control it. Its movements aid in chewing and swallowing, plays a really important role in speech, and it also has a sensory role. Okay, that's where your taste buds are, to help detect what you've just put in your mouth. Although what has a larger ability to detect what you've just put in your mouth? Your tongue or your nasal cavity? Your nasal cavity. True, your eyes. Your eyes help detect too. Hey, so here we've got human dentition, which is responsible for chewing or mastication. Okay, and humans have 8 incisors, 4 canines, 8 premolars, and 12 molars. Okay, so our dentition sort of says that we are omnivores. Okay, we've got, our canines are not really robust, but we've got canines, right, to help rip flesh. Okay, so we can be meat eaters, but we also have these really nice molars for grinding things, so we can also be plant eaters. Right, so chewing, the process of chewing helps grind and break food into smaller pieces so that you don't choke when you swallow. Right? So as a kid, your mom was always telling you to just chew your food, right? So that you didn't choke. It also increases the surface area for the salivary enzymes to act on. It also burns calories, right? So people have done studies where crunchy, chewy food burns more calories than food you just let slide down the back of your throat, like those Go-Gurts, right? And those applesauce squeezy packets, the baby food for, grade school kids now. Right, you want to mix your food with saliva so that those salivary enzymes can work, but you also have other things in your saliva that you want your food to interact with. And then chewing also helps stimulate your taste buds. Okay? And here are our salivary glands. We've got parotid glands, sublingual glands, and submandibulary glands that produce our saliva. Right, notice we've got our guy chewing. So One of the reasons we're also omnivores is because our jaws can go up and down really well as side to side for grinding. So if you think of your dog and cat, which are much more carnivorous, their teeth do not go side to side, right? They go up and down. That's how their jaws work. So they have much bigger bite forces than we do. But they can't really grind things up, okay? So when you give your dog a rawhide, it likes to swallow it, let it stew a little bit, regurgitate, at least my dog does. Try again. Okay, so saliva is produced by your salivary glands, and it's primarily water. Okay, so this is our first secretion into the GI tract, is saliva into our mouth. And it is primarily water, but 0.5% is electrolytes, okay, so some bicarbonate in order to neutralize any acid you've taken in, as well as some proteins. And those proteins are amylase, which is a digestive enzyme, okay, some mucus to help lubricate the food so you can swallow more easily, and then some lysozymes, okay, to help destroy the bacteria that is coating your food. Okay, so this is your first time trying to destroy that bacteria that's coating the food. Okay, so amylase starts carbohydrate digestion. So carbohydrate digestion starts in the mouth. Okay, so in lab you had to chew those saltines in order to get saliva production. You didn't chew any? Last week you did digestion? Oh, you haven't done the digestion. Next week in lab, you'll have the chance to chew on unsalted saltines. Delicious. Okay? If you chew on them for long enough, they start to taste sweet, right? Because the amylase starts breaking down the starch that's in those saltines into glucose so that you actually detect a sweet sensation. Yes?
Do they actually make those, or do you have to like special order them?
Unsalted saltines? Yeah, they make them. Lots of people have to have low salt diets. We don't special order them. You ain't at the grocery store.
Why would you buy saltines if you're not supposed to have salt?
Because you can buy unsalted ones. I don't know. So crumble into your soup, right? For textures. Animal crackers.
It's kind of like why do they make vegetarian bacon?
Anyway, you can even chew salted saltines for long enough and they will taste sweet. It's not the salt that's disrupting the sweet sensation. It's that you have to chew them for long enough for amylase to break down those complex carbohydrates into disaccharides or monosaccharides, which is what your taste buds detect as the sweet sensation. Okay? You need your food to be lubricated so that you can swallow it. Okay? And if you eat something dry, you want it to be well lubricated and moistened to swallow it. Okay? So if you're snacking on potato chips right now, you want to chew them up and secrete a whole bunch of saliva so that you can swallow them, so they don't scrape all the way down your esophagus. Okay? You want to kill any bacteria, so lysozymes help do that. Okay? And then saliva also helps rinse away material from your teeth. Okay? So that you don't have tooth decay. So people who have chronic dry mouth often have to rinse their mouth frequently with mouthwashes, okay, because they are really susceptible to tooth decay. Okay, that water acts as a solvent so that those disaccharides, amino acids, the sodium, etc., can hit the taste buds so you can detect whether you're eating something sweet, salty, sour, bitter, or umami. Remember our 5 principal tastes? You need saliva for speech, right? If you have a really dry mouth, it's really hard to speak. So nervous speakers oftentimes don't produce enough saliva, and so you'll see them taking sips of water. Okay. Keeps your mouth and teeth clean, and it's also rich in bicarbonate buffers in order to buffer anything acidic you take it, because also decreasing the pH in your mouth increases tooth decay. How many people want to go into dental school? These couple of slides were for you guys.
Thank you.
Yeah. Sorry, that's it though. Okay. So swallowing is a reflex. Okay, so when you go to swallow food, you're going to push the bolus of food, which is being shown in green, with your tongue to the back of your mouth. And that's going to initiate the swallowing reflex. And during the swallowing reflex, the epiglottis moves downward to cover the glottis. Okay, because you want to block passage into the trachea, right? Because if food enters the trachea, what happens? If you choke, you die. Yep. Okay, so that's when you're choking, when food or water enters your trachea. Okay, so you want to block the trachea, and at the same time, you want your upper esophageal sphincter to open so that food can enter your esophagus. Okay, so the swallowing reflex causes the epiglottis to cover the glottis so that you don't choke, and then the upper esophageal sphincter opens, and then that bolus of food is going to enter your esophagus, and then you're going to start peristalsis. Okay, so muscle's going to contract and move that bolus of food all the way down to the stomach. Okay, once that bolus of food hits the lower esophagus, the lower esophageal sphincter will open. Okay, and why do you think you have a sphincter at the stomach? What's the stomach filled with? Acid. So what happens when your lower esophageal sphincter isn't tight enough and you get some acid up into your esophagus? Acid reflux, which often people call heartburn, right? So when your esophagus gets burned by that acid coming from your stomach, it has nociceptors. However, you don't have a map in your somatosensory cortex for your esophagus, right? It gets referred to the skin surface over your heart. So that's why it's called heartburn. Okay, you're actually damaging your esophagus. It gets referred to your skin surface over your heart. So you're not having a heart attack. Okay, you just ate food that was too spicy, or you ate too fast, or you're too full. Okay, pharynx is also known as your throat. It's the common passageway for both respiratory system and GI tract. Your esophagus is a muscular tube. No digestion, no absorptions occurring in your esophagus. It is just a passageway from your mouth to your stomach. Okay, the upper 1/3 is skeletal muscle. The lower 2/3 is smooth muscle. Okay. You've got your upper esophageal sphincter, which is skeletal muscle, but you really don't consciously control it. It's part of the swallowing reflex to open it and then close it. Okay, and it prevents air from entering your GI tract when you're breathing. Okay, the lower esophageal sphincter is completely smooth muscle, no conscious control, and it separates your esophagus from your stomach, and it keeps the stomach acid in your stomach so that you don't damage your esophagus. Okay, so our food's now entered our stomach. Any questions about the process of getting it there? You guys do it all the time, so it's easy. You chew, you swallow. Okay, so let's talk about what happens in our stomach. So your stomach is J-shaped. It's this big sac that lies between your esophagus and your small intestine. And it has 3 sections. You've got your fundus, okay, you've got the body, and you've got the antrum. And the 3 main functions of your stomach, one is just to store food. Okay, your stomach acts as a storage organ. When it's empty, it's like 50 mils. But it can extend to hold 5 liters. And if you train to be a competitive eater, it can get even larger. Okay, so competitive eaters train, right? They try to fill their stomachs full of bulk on a regular basis so that it stretches. And that's why oftentimes the best competitive eaters are actually not really obese, right? Because that layer of fat actually prevents your stomach from fully extending. So the Coney Island hot dog champ is always a really small guy. Okay. So competitive eaters have to train, so they're considered athletes. Right. The reason why your stomach is storing food is because you don't want to dump your entire meal into the small intestine at once, because digestion and absorption takes time. Okay? And the more protein and fat that was in your meal, the longer it stays in your stomach. Right? So that's why diets high in protein and fat have higher satiety. And satiety means how full you feel. Right? So if you just eat, a bunch of even salted saltine crackers, you're probably going to be hungry pretty soon afterwards because it's just carbohydrates. Right? They pass through really quickly. Whereas protein and fat are going to stay in the stomach for longer because they're harder to digest and absorb. Right? So that's why there's that whole protein water craze. The idea is that it might make you feel full, right? May give you a sense of satiety. I don't know if it actually works. Hey, your stomach secretes hydrochloric acid and enzymes to begin protein digestion. Okay, so carbohydrate digestion starts in the mouth, but then it stops in the stomach because that amylase is a protein and it gets digested. Protein digestion starts in the stomach. That low pH, the hydrochloric acid, activates the proteolytic enzyme. Okay, so one of the reasons for the low pH is to activate the enzyme in your stomach that does protein digestion. And the other reason is to kill any bacteria on your food. Okay, we have pretty low pH stomachs, but carnivores have even lower pH stomachs. So that's why you can feed your dog and cat raw chicken and they don't get salmonella. Right? Because their stomachs actually have even lower pHs. Right, and then our stomach churns things. Okay, acts like a big butter churner. All that mixing movement, our stomach is super muscular, and we're basically pulverizing food or liquefying it, and then it's called chyme. Okay, so if you don't chew your food well, your stomach's going to pulverize it, completely liquefy it, and then that point is called chyme. 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 Yeah.
So I'm just curious. So eating crunchy food.
Yeah.
So if you swallow large bits of food without chewing, would that mean your stomach is going to burn more calories, pulverizing it into?
I don't know about that. Yeah. I mean, to crunch a potato chip, your stomach to crunch a potato chip doesn't take much, right? Because at that point, you've got a whole bunch of liquid in there as well. But yeah, I mean, you have to get a bunch of people to swallow potato chips whole.
Yeah, no one's going to volunteer to do that. It hurts.
Right, as it scrapes down your esophagus. Yeah.
I was thinking more like a whole grape.
A whole grape. Oh.
It's not crunchy, but it is chewy.
It is chewy. That is true. Yeah. Yep. Okay. Although some things just don't even get pulverized. So corn, right? Okay. Yeah.
Kind of a dumb question, but why does our stomach growl?
Oh, our stomach growls because it's empty, right? And it's starting to do those muscular contractions, right? But there's nothing in there. Okay, so it's getting ready for a meal because maybe that's the time you normally take in a meal, or you've smelled something that smells delicious, okay, and you've gotten your GI tract ready to go, but nothing's actually entered your stomach yet. Yep, so it's doing all those mixing and pulverizing movements without anything in your stomach. Okay, your stomach has two sphincters, the lower esophageal sphincter to protect the esophagus from gastric acid, and then it has the pyloric sphincter to protect your small intestine from gastric acid. Okay, so the pyloric sphincter serves as the barrier between the stomach and the small intestine because chyme is super acidic. Right? And you only want to release a little bit at a time into the small intestine because otherwise you will erode your small intestine. Okay? So release only a small amount of chyme at a time through that pyloric sphincter. Okay. Okay, so we were talking about the stomach, right? And how the stomach serves as a storage organ. It's super distendable, right? Because you want to give time for your stomach to start protein digestion. Okay, so your stomach has a number of secretory products, right? One of those secretory products is pepsinogen. One is hydrogen ions, one is intrinsic factor, one is gastrin, and then of course loads of mucus. Okay, so here we have a cross section of your stomach. And those secretory products are coming from cells that line your gastric pits. Okay? Basically, the interior of your stomach is this superacidic environment. So one way to protect your cells from that superacidic environment is to place them inside these pits and then have this really thick layer of mucus that's covering the pits. Okay? And so you've got these different types of cells lining your gastric pits. You've got neck cells, which are towards the top, and they're the ones that secrete the mucus that is protecting your stomach lining. Okay, and then we have chief cells, and we've got parietal cells. So the parietal cells are the purple ones. The chief cells are the ones that are whatever color you want to call this. I don't even know what to call that color. Off-white? Okay. So you've got way more chief cells. Take-home message. We have way more chief cells than you have parietal cells. Chief cells are what secrete pepsinogen. Pepsinogen is an inactive proteolytic enzyme. Proteolytic enzymes break peptide bonds. Remember, peptide bonds are the covalent bonds holding amino acids together. Okay, so pepsinogen is an inactive proteolytic enzyme. It gets activated by low pH. Okay, so pepsinogen is secreted into the gastric pits, and then once it hits the lumen of the stomach, it's going to be activated by that low pH environment. And then it's going to start to digest your protein. Okay? The reason why it's secreted in an inactive form is that you don't want to digest the protein inside those chief cells. Okay? So they secrete it in an inactive form. It gets activated by hydrogen ions. Okay? The hydrogen ions released by the parietal cells help maintain the acidic environment of the stomach, which activates pepsinogen. Okay? And it helps to protect you from bacteria. Okay, so it destroys bacteria that was in your food. And so that really nasty E. coli 0157, that causes a lot of foodborne illness, it evolved in the stomach of cows that got fed a whole bunch of corn, which made their rumens, which are normally pretty neutral pH, low pH. Okay? And cows have these giant stomachs that are just filled with bacteria. Most of them we kill off in our stomach, no problem. But that 0157 can survive in a low acidic environment. Right? And then therefore it doesn't get destroyed in the stomach, enters our small intestine. If it gets into your bloodstream, can cause kidney failure, etc. Okay? Your stomach also secretes what's called intrinsic factor. So your parietal cells secrete acid as well as intrinsic factor. Intrinsic factor is necessary for absorption of B12. And there's a genetic disorder where people don't produce intrinsic factor. Right? And they suffer from pernicious anemia. You guys remember that pernicious anemia was anemia due to lack of B12, which you need for cell division, DNA replication. So they have to get injections of B12. Because no matter how much B12 you feed them through their GI tract, they don't absorb it. Okay, so they have to get injections of B12. It's super uncommon. Okay. G cells, which aren't being shown here, but they're also in the gastric pits, secrete gastrin. And gastrin is a hormone, right, that regulates pepsinogen secretion and acid secretion. So the more amino acids that are in the meal you took in, the more gastrin your stomach releases. Okay, so your stomach can detect what is in the food you just put in it. And if it's a lot of protein, it wants to hold on to it for a while. Okay, and it wants to secrete a load of acid and a load of pepsinogen in order to start digesting that protein. Okay, gastrin then also signals to the small intestine that we have a high protein meal. Okay. Then, of course, these next cells are going to secrete the mucus that helps protect the stomach lining. Hey, any questions about the stomach? All right, so our next organ on our trip from the mouth to the anus is the small intestine. Okay, your small intestine is 8 to 10 feet long, right? And we're going to separate it into 3 segments: the duodenum, the jejunum, and the ileum. Okay, so the stomach empties into the duodenum, which empties into the jejunum, which empties into the ileum. It's just a big coiled mass right here. The ileum enters into the large intestine. Okay, your small intestine is the site of digestion of all 3 major macronutrients. Okay, so carbohydrates, proteins, and fat, as well as the absorption of all 3 macronutrients. Okay, your small intestine does some secretion. Okay, this juice, right, that's secreted by the small intestine doesn't have any digestive enzymes. Okay, all the digestive enzymes that are in the lumen of the small intestine actually get secreted by the pancreas. Okay, so your pancreas secretes digestive enzyme into the small intestine. The small intestine itself doesn't secrete any digestive enzymes. It does secrete some salt and mucus solution, right? The enzymes that your small intestinal cells make are brush border membrane enzymes. Okay, so our epithelial cells lining our small intestine are called enterocytes. And like those proximal tubule cells, the apical membrane that faces the lumen of the small intestine, right, it has microvilli. And those microvilli look like a brush. So it's also called the brush border. Okay, so the apical membrane with those microvilli is also known as the brush border. So the digestive enzymes that these enterocytes produce are membrane bound. So they stick them on the microvilli. Okay, so the nutrients have to come really close to the cells before the enzymes will degrade them. Okay, the digestive enzymes that your small intestine produces, one is enterokinase. Enterokinase converts trypsinogen, which is a proteolytic enzyme that's inactive, that gets secreted by the pancreas. Okay, so the pancreas secretes trypsinogen into the duodenum, and then those enterocytes convert trypsinogen into trypsin. And then trypsin continues protein digestion. Hey, you also have a series of disaccharidases that are brush border enzymes. And disaccharidases convert disaccharides, which are 2 monosaccharides held together by a covalent bond, into monosaccharides. So you guys all produce sucrase as a brush border enzyme. And sucrase digests sucrose into glucose and fructose. Some of you still produce lactase, right? And lactase is a disaccharidase that digests lactose, right? That converts glucose converts lactose into glucose and galactose. If you are lactose intolerant, it's because you don't produce lactase anymore. Okay, and then the other class of brush border enzyme are the aminopeptidases. And these convert peptides, or short chains of amino acids, into amino acids. Okay, so monosaccharides are what get absorbed, amino acids are what get absorbed. There are no brush border enzymes involved in fat digestion. Okay, and we'll talk about why. Right, your pancreas releases a series of enzymes that digest things like protein in the case of trypsin. right? Or carbohydrate in the case of pancreatic amylase, or fat in the case of pancreatic lipase. Your pancreas also secretes a boatload of bicarbonates. And why does it secrete a bunch of bicarbonate? What's that bicarbonate going to do? It's going to buffer, right? Because the stomach is releasing super acidic chyme into the small intestine. And your small intestine is not heavily protected the way your stomach is, because you want absorption to occur in the small intestine. So you don't want the super thick mucus layer. So you need to buffer that super acidic chyme as quickly as possible. And you do that by releasing bicarbonate. Right. The other accessory digestive organ that releases a secretion into the small intestine is your gallbladder. Your gallbladder releases bile into the duodenum. And what does bile contain that helps with digestion? Who knows? What does bile help you digest? Fat. Okay? So you have bile salts in the bile that help emulsify fat. Yeah. Yeah.
On this slide, I just want to make sure I have this right. The small intestine does not secrete any digestive enzymes.
Nope. All the digestive enzymes in the lumen, so the free-floating ones, are secreted by the pancreas.
And that includes the brush border enzymes?
No, the brush border enzymes aren't free-floating. They're attached, so they're not technically secreted.
Oh, I see. So
Right?
there are digestive enzymes.
Made by the enterocytes, but they hold on to them.
Oh,
They're membrane bounds.
okay.
Okay.
So it does make digestive enzymes, they're just not free.
It makes them, it just doesn't secrete them.
Okay.
Yep, it holds on to them. They're brush border, meaning they're membrane bounds. Okay. Right. The major role of the small intestine is digestion and absorption. Okay, so digestion is due to pancreatic enzymes as well as those brush border enzymes. Okay, so pancreatic enzymes continue carbohydrate digestion. And we say continue because where did carbohydrate digestion start? In the mouth. Okay? So carbohydrate digestion started in the mouth due to salivary amylase. What happened to the salivary amylase once it hit the stomach? It was denatured and digested. Okay? Pancreatic enzymes continue protein digestion because where did protein digestion start? In the stomach. Okay? Trypsin, which is the pancreatic proteolytic enzyme, works best at a neutral pH. Pepsin, which is the stomach's proteolytic enzyme, works best at a low pH. So as soon as pepsin hits the small intestine, it actually gets denatured, and then it gets digested by trypsin. Okay. Brush? Yeah.
So when our food enters the small intestines, from what you're saying, I get the impression it's been completely broken down from its original state as food, and it's just kind of this.
It's been liquefied.
Yeah.
It hasn't been broken down in terms of covalent bonds being broken, except for some with carbohydrates due to amylase in the mouth, and some of the proteins have had peptide bonds broken due to the pepsin.
But it doesn't look like meat and vegetables. I mean, it's just kind of.
It's a slurry.
Okay.
Yeah. Okay. It's a super acidic smoothie. Okay. Except for corn, of course, right? Which still looks like corn even after it's been passed completely through the GI tract.
Why can't we break down corn?
Oh, it's because of the
Shell?
the shell. Yeah.
Just some. We don't make the enzymes or whatever.
Nope.
No.
Yep. Okay. All right. Brush border enzymes complete carbohydrate and protein digestion. Okay. Fat is only digested in the small intestine. And the digestive enzyme that digests fat is a pancreatic lipase. There is a lipase secreted in saliva. It's called linguinal lipase, but it plays a super minor role in lipid digestion. Okay? Fat is entirely digested within the lumen of the small intestine by pancreatic lipase. Okay? There's no brush border enzyme involved in fat digestion. Okay, absorption. Small intestine absorbs almost everything presented to it. And the vast majority of absorption occurs in those upper two segments, the duodenum and the jejunum. Okay, the reason why the small intestine is so good at absorption is because it has a huge surface area. Okay, so it has permanent circular folds, so it's sort of like squeezed together, like a xylophone, right, in those circular folds. And then it has finger-like projections called villi, and on those villi is where you get the enterocytes, and then the enterocytes have microvilli. Right? So this greatly increases surface area so that you can get almost full absorption of everything you present to the small intestine. Okay, so here, we don't see the circular folds in here very well, but we do see the villi. Okay, so here are those villi, and on the villi, so here we have a blow up of a single villi, we have the enterocytes, and the enterocytes have microvilli on their brush border. So this is the apical membrane that's facing the lumen of the small intestine. This is the basolateral membrane. Okay, once things get absorbed across those enterocytes, each one of those villi have a capillary bed associated with it. Okay, so all the carbohydrates and amino acids that get absorbed across the microvilli through the basolateral membrane are just going to enter the bloodstream. Okay, the dietary fat that gets absorbed, remember it gets packaged inside the enterocytes into those chylomicrons. They're too big to get into the capillary bed, and they enter the lacteal instead. So this green thing is the lacteal. So each villi has a lacteal and a capillary bed associated with it. Okay. These microvilli, or enterocytes will get sloughed off. Okay. And so your villi are constantly replacing those enterocytes. Okay, they replace them from the base upward. So as the ones on the tip get sloughed off, new ones are there to replace it. Okay, so what we're going to do is go through the major macronutrients, okay, and how they're digested and absorbed. So we're going to start with carbohydrates, because who doesn't love carbohydrates? Okay, hopefully you ate a lot of Easter candy yesterday. Okay, if you ate a lot of Peeps, all you ate was carbohydrates. Okay, the typical American diet is anywhere between 250 to 800 grams of carbohydrates. Okay, depending on how much you love bread. Okay, most is consumed as disaccharides or polysaccharides. So the major disaccharides in our diet are sucrose, that's table sugar. Okay, that's that white granulated sugar you can buy in 5-pound bags. Okay, lactose is milk sugar. Okay, maltose is 2 glucose monomers held together by a covalent bond, and it's the product of fermentation. Okay. Sucrase breaks down sucrose. Lactase breaks down lactose. Maltase breaks down maltose. Okay, so ASE at the end means it's the enzyme. OSE means it's the disaccharide. Okay, the polysaccharides, or the complex carbohydrates in your diet, are starch, okay, glycogen if you eat a lot of meat, because glycogen is the storage form of carbohydrates in animals. Okay, starch is the storage form of carbohydrates in plants, right? And then cellulose. So cellulose is your dietary fiber that we do not digest or absorb in our small intestine. Okay, we do not have cellulitic enzymes. Dietary fiber goes into your large intestine. Yeah?
The last three are polysaccharides?
The last three are polysaccharides, long chains of glucose. We only absorb monosaccharides. Okay, so if you are lactose intolerant, you do not produce lactase anymore. Okay, you only produced it in probably like the age of 2 or so, right? Because humans used to only drink milk until they got weaned, right? People of Northern European descent evolved the ability to produce lactase into adulthood, right? Because they were raising a lot of cattle and sheep and goats and drinking their milk and eating cheese, right? And so if you can drink milk and eat cheese without horrible diarrhea, stomach cramps, and gas, then you produce lactase, okay? Yeah.
So I've heard of people being like lactose sensitive.
That's the same thing. Lactose sensitive is the same thing as being lactose intolerant, right? If you eat a load of lactose, bad things happen inside your stomach and small intestine and large intestine, okay? So if you don't digest the lactose, the bacteria in your large intestine do, and it sucks in a bunch of water, right? It causes them to go crazy with fermentation because they find it super easy to digest, which leads to high gas production. So that's where you get the cramping and the diarrhea and the gas production because your bacteria in your large intestine have no problem digesting the lactose. Okay. Right? Because you can't absorb lactose. You can only absorb the glucose and galactose that lactose is made up of. Right, so here we have a polysaccharide. So this is starch. It's long chains of glucose. Each one of these little green hexagon is a glucose molecule. Okay, and it's branched. And so if you had some potatoes with Easter dinner, okay, you took in a whole bunch of starch. If you chewed those potatoes well enough, salivary amylase started to digest them, break down the covalent bonds into limit dextrins, which are just smaller chains of glucose, or into maltose. Okay? Yeah?
What's the difference between glucagon and starch if they're both?
You mean glycogen?
Yes.
So glycogen and starch, glycogen's also long chains of glucose. It's just a slightly different branching pattern.
So it's just the branching pattern.
It's just the branching pattern. But glycogen is highly digestible as well. Okay? You usually don't take in huge amounts of glycogen, right? Whereas you can take in a huge amount of starch all at once. Okay? Because remember, plants like to store energy as carbohydrates because they can be really heavy. They don't have to move. Whereas animals like to store energy in the form of fats. Okay? Because it's lighter. All right. So if you don't chew your food very well, in the small intestine, pancreatic enzyme will do the same thing. It'll break the starch down into limit dextrins and into maltose. Okay. Then you have brush border enzymes on those enterocytes to finish the digestion of carbohydrates. So carbohydrate digestion starts in the mouth. Okay, then pancreatic amylase will convert any remaining starch into limit dextrins and maltase, and then dextrinase will convert those limit dextrins into glucose, and then glucose will be absorbed. Okay, you also have glucoamylase, so any polysaccharides that either one of the other amylases haven't digested, pancreatic or salivary, also will convert it into glucose. Okay, sucrase will convert sucrose into fructose and glucose. Lactase will convert lactose into galactose and glucose. Maltase will convert maltose into 2 glucoses. Notice we have monosaccharides as the end product. Okay? These brush border enzymes are on those enterocytes. And so the reason why those enterocytes don't secrete the brush border enzymes into the lumen is because as soon as they form the monosaccharides, they want to make sure those monosaccharides are right there to be absorbed, right? Because if you form that glucose inside the lumen of the small intestine, there's a chance it's not going to hit one of those enterocytes. Whereas if you form the glucose right at the microvilli, chances are you're going to absorb it. Okay, so make the product that's going to be absorbed right where it's going to be absorbed. Okay. Absorption of monosaccharides in the small intestine is exactly the same as absorption or reabsorption of glucose from the proximal tubule. Okay, so absorption of glucose and galactose involves secondary active transport across the apical or brush border enzyme via SGLT1, right? And then facilitated diffusion across the basolateral membrane. And we'll look at it again, but it's exactly the same as how glucose got reabsorbed from those proximal tubule cells. Same transporters. Okay, fructose gets transported by facilitated diffusion across both the apical and basolateral membrane, which we didn't talk about fructose absorption when we talked about kidneys. Okay, so here we have an enterocyte lining the small intestine. So here's the lumen, here's that brush border or apical membrane, here's the basolateral membrane that's facing the internal body compartments. Okay, so on the apical membrane, we have the brush border enzyme. So we're going to use maltase as the example. Maltase is taking the disaccharide maltose and breaking that covalent bond between the two glucoses. So it's converting maltose into two glucoses right at the apical membrane so that glucose can be absorbed right away. Okay, like any cells, you have high sodium outside and low sodium inside of the cells. So this transporter, which is known as the sodium-dependent glucose transporter 1, or SGLT1, what it does is it lets sodium in down its electrochemical gradient, and sodium so badly wants to enter the enterocytes that it pulls glucose up its concentration gradient. Okay, so you have high glucose inside the cells, low inside the lumen, because most of the carbohydrate inside the lumen of your small intestine is either disaccharides or limit dextrins or starch. Okay, because as soon as glucose gets formed, it gets actively absorbed. And then we have high glucose inside the enterocytes, and we have low glucose in those interstitial fluid. Because as soon as glucose hits the interstitial fluid, it gets picked up by those capillaries that line all the villi, and it gets whisked away to the liver, right? So that means low glucose in the interstitial fluid. So that means to cross out of the cell, it can go by facilitated diffusion. And these transporters are called GLUT transporters. So the GLUT transporters are all facilitated diffusion. Okay. In order to maintain the sodium gradient so that the secondary active transporter works, we have loads of sodium-potassium pumps, which are primary active transporters on the basolateral membrane. Okay, so just like those proximal tubule cells, you get sodium-potassium pumps only on the basolateral membrane, and then all sorts of secondary active transporters on the apical membrane. Okay. Any questions about carbohydrates? Okay, let's talk a little bit about proteins then. So the typical American diet has 125 grams per day of protein. And we're a meat-eating society, so we take in more protein than we actually need. We only require about 40 to 50 grams, right? But we love to eat our meat in the U.S., okay? Protein that is digested and absorbed from your small intestine, some of it came from what you put in your mouth, okay? This is exogenous protein, came from outside of your body. Some of it is endogenous, meaning it came from inside of your body. So that means might be the digestive enzymes that got secreted into your GI tract. Okay, you're going to digest those and reabsorb the amino acids, or any of the protein in mucus, okay, or any of the protein in the cells that got sloughed off. So you remember I said those enterocytes will get sloughed off, especially when they're on the tip of the villi. When they get sloughed off, you digest them and reabsorb the monomers. Okay, so you digest and absorb both exogenous protein and endogenous protein. Okay, in order to be absorbed, proteins have to be digested into amino acids, dipeptides, or tripeptides. So unlike carbohydrates, you can absorb 2 amino acids held together or 3 amino acids held together. Okay? Carbohydrates, only monosaccharides get absorbed. But you do have some transporters that can do 2 amino acids together, so dipeptides, or 3 amino acids together, which are tripeptides. Okay? And the enzymes that break the peptide bonds holding amino acids together are known as proteases or proteolytic enzymes. Okay? They're synonyms. So proteases are proteolytic. Yeah?
If we need so little protein, what's the point of those protein shakes?
Exactly.
Oh, okay.
To make money.
I mean,
Yeah.
from the sound of it, you would have to.
So your protein requirements might be higher if you are trying to build muscle,
Right.
right? But you probably just don't need as much that is in a protein shake. Okay.
So, is that just going to make you gain weight?
Yeah, so you can convert excess amino acids into fat,
Right.
right? If you take in too many amino acids, you will convert them into fat to store that energy, right? So drinking protein shakes doesn't make you bulk up unless you also do exercise along with it to make you bulk up.
If you're taking a bunch of those protein shakes and you're not really working it, you're just going to start getting bigger.
Right, bigger in the way you didn't want to get
Right.
bigger. Right? And too much protein can also really work your kidneys, right? Because you have to, when you convert protein into fat, you have to take off that amino group. And then you have to excrete that amino group as urea, right? So, you know, if you don't need a high protein diet, there's no reason to eat a high protein diet.
So then,
So if you're not a bodybuilder.
does that mean that we just, in our average, are hurting our kidneys just with our...
No, this isn't so high that you'd actually hurt your
Oh.
kidneys. Yeah. This is just excess protein, more than you actually need.
Okay.
Yeah. Yep. Okay. Right, so protein digestion starts in the stomach with the proteolytic enzyme pepsin. Okay, so remember those chief cells secrete pepsinogen, and then the parietal cells secrete hydrochloric acid, which converts pepsinogen into pepsin. Okay, so pepsinogen is inactive until it experiences a low pH environment. Then it gets activated. Then it's going to start breaking peptide bonds in the protein inside your stomach. So you'll form smaller peptide fragments. Pepsin works best at a low pH. Okay, so as soon as it hits the small intestine, it denatures. Okay, and then it will be digested. So it's one of the sources of endogenous protein that gets digested. Okay, in the small intestine, your pancreas releases proteolytic enzymes, one of which is trypsinogen. Okay, trypsinogen is inactive, and it gets activated by the brush border enzyme enterokinase. So here we've got the enterocytes lining the small intestine. Okay, so the pancreatic duct dumps its secretory products right into the duodenum. Okay, and then that trypsinogen, which is inactive, immediately gets activated by enterokinase, and then trypsin activates chymotrypsinogen, right, into chymotrypsin, and then it activates procarboxypeptidase into carboxypeptidase. Okay, and then trypsin also directly breaks peptide bonds in exogenous protein as well. Okay, so not only is trypsin a proteolytic enzyme for exogenous protein, it also activates 2 endogenous proteolytic enzymes. Okay, and then you also have aminopeptidases on the brush border membrane. Okay, so if you printed this out, you could add another brush border enzyme, which are the aminopeptidases, which would start breaking down those peptide fragments that are being made by trypsin, chymotrypsin, and carboxypeptidase. Okay, amino acids cross the apical membrane via secondary active transport that is sodium linked, so very similar to how glucose gets absorbed across the apical membrane, or by facilitated diffusion, depending on what amino acid it is. Okay, so the amino acids that are essential, that we can't synthesize ourselves, usually get absorbed by secondary active transport, okay, because you want to absorb all of them. And then the ones that aren't essential because we can make them, so like glutamate, right, would move by facilitated diffusion across the apical membrane. All amino acids cross the basolateral membrane by facilitated diffusion. Di and tripeptides cross the apical membrane by active transport. And then inside the enterocyte, they get broken down to amino acids. And then they just move across the basolateral by facilitated diffusion. Okay, so here we have a nice cartoon showing the whole process of protein digestion. So exogenous protein and endogenous protein. Okay, so dietary protein as well as any digestive enzymes, protein in epithelial cells that are sloughed, leaked plasma proteins. Your small intestine and stomach doesn't care. Protein is protein. Whether it's endogenous or exogenous, it's going to break it down by proteolytic enzymes. Pepsin in the stomach, the pancreatic proteolytic enzymes, trypsin, chymotrypsin, and carboxypeptidase. Okay, those are the pancreatic proteolytic enzymes. They're going to break down protein into small peptides or amino acids. Small peptides will be further broken down by aminopeptidases, okay, and then they're going to be absorbed across the apical membrane by either secondary active transport, okay, or in the case of di and tripeptides, active transport, okay, directly using ATP. Those di and tripeptides, once they're inside the enterocyte, they're going to be broken down by intracellular peptidases into amino acids. The amino acids are just going to be amino acids, and no matter where they came from, amino acids cross the basolateral membrane by facilitated diffusion. Once across the basolateral membrane, they're going to enter those capillary beds, and they're going to be whisked away to the liver. Okay. Any questions about protein digestion? Right. The last macronutrient we're going to talk about is lipids. Okay, and lipids pose a problem. Lipids are not water soluble. Okay, so they don't want to mix with that liquid chyme or the liquid intestinal content. Right? And they hang out together, they congregate into these large fat droplets. And digestive enzymes are all proteins, and therefore they are water soluble. Okay, so if you have these big fat droplets, lipases, which are the digestive enzymes, can't get into the interior. So they can only work on the exterior, which is not very effective. Okay, so you need to emulsify dietary fat in order to get efficient digestion and absorption. Okay. Typical American diet, huge variation in fat, okay, from 25 to 160 grams of lipids. Whether you eat a lot of fat or a very little bit of fat, most of the fat you take in is triglycerides. Okay. Some of it is phospholipids, so from plasma membranes of both plants or animals that you're consuming, and very little of it is steroids, right, or cholesterol. Vast majority is triglycerides. Okay, the enzymes you have for digesting fat are lipases, and the major lipase is secreted from the pancreas. Okay, lipases are proteins. Proteins are water soluble, so they can only work at the edge of the fat droplet. So we have to emulsify our dietary fat, and bile salts do that. Okay, so the role of bile salts is to emulsify your dietary fat. Your bile salts do not digest dietary fat. They emulsify it so that your lipases can digest it. Okay, so your gallbladder stores bile. The bile is ultimately made by your liver. Okay, so your liver makes bile salts. Your gallbladder stores those bile salts, and whenever you eat a fatty meal, your gallbladder contracts and squirts out bile that contains a bunch of bile salts, and then those bile salts emulsify your dietary fat so you can digest it and absorb it. Okay, so here we've got bile salts, and bile salts are synthesized in your liver from cholesterol. Okay, so here's the cholesterol containing portion. The cholesterol containing portion is hydrophobic. Okay, here, look at this. We've got sodium that's charged. This portion is hydrophilic. This means bile salts are amphipathic. What other molecule did we talk about that's amphipathic? Phospholipids. Okay, so it's both polar and nonpolar at the same time. So this means the nonpolar hydrophobic side will interact with your dietary fat. And then the polar side will interact with the liquid inside your small intestine. Okay, so it's emulsifying your fats, breaking those really big fat droplets into much smaller ones, which increases the surface area for digestion to occur. Okay, so without bile salts, you would just have these giant fat globules. Okay, but luckily you'll secrete bile salts into the small intestine, and they will interact with those triglycerides, right, or phospholipids, and then the hydrophilic side will interact with the watery digestia, so you'll get these fat globules turning into smaller fat droplets. And this increases surface area for lipase to work, because lipase is water soluble, so it's only going to work on the edges. Okay? So bile salts emulsify your dietary fat. Okay. Lipases digest your dietary fat. So they take triglycerides, which are a glycerol with 3 fatty acids, and convert it into monoglycerides plus 2 fatty acids. Okay, if this occurs really close to an enterocyte, they'll just cross the membrane by diffusion, right? Because they're fat soluble. So they'll just cross into the enterocyte. If this digestion by lipase occurs in the lumen of the small intestine, those released monoglycerides and fatty acids are going to congregate together. And when they congregate together, they're called micelles. Okay, so micelles are groups of digested fat products, so monoglycerides and fatty acids, congregating together, waiting to bump up against your enterocytes and be absorbed. Okay, so we have the big fat droplets that get, or big fat globules that get emulsified by bile into fat droplets. The fat droplets get digested by lipase, and those products form micelles. Okay, so absorption is pretty easy. To get across the apical membrane, they just have to come in contact with it because they're fat soluble. They just diffuse across. However, once inside the enterocyte, fat gets packaged. Because if you just dumped a whole bunch of fat into your bloodstream, then you would have big fat globules hanging out in your bloodstream, which is not good. Okay, so you need to package fat with protein so that it can be transported in the bloodstream. Okay, so fat likes fat. So any fat that gets absorbed across the apical membrane is going to associate with the fattiest portion of those epithelial cells, which is the smooth endoplasmic reticulum. So remember way back when we talked about cells, the smooth endoplasmic reticulum makes fat. Right? So that's where the vast majority of fat is in your cells. So that is going to draw any absorbed fat to it. And then the smooth ER is going to reform the triglycerides or other lipids. Okay? And then it's going to send those to the Golgi apparatus or Golgi complex. And it's your Golgi apparatus that packages your dietary fat into chylomicrons. And chylomicrons are just protein and fat together. They're now really big. Okay? And because there's now protein associated with that fat, it can't just diffuse across the plasma membrane. So chylomicrons get released across the basolateral membrane by exocytosis. And they are so big, they can't get into the capillary beds, so they enter those lacteals instead. Right, so let's look at this as a cartoon. Okay, so if you wanted to, you could draw a giant fat globule up here, right? Like a moon blocking out the sun. Okay, and then you would have bile salts. Okay, and bile salts are emulsifying those big fat globules into fat droplets. So here's our bile salts hanging out. Okay, that's increasing the surface area for lipase from the pancreas to digest things like the triglyceride. So here's lipase, digesting that triglyceride into monoglyceride and fatty acids. Okay, those fatty acids and monoglycerides can get absorbed right away if they're close enough to the enterocyte, or they're going to congregate together into micelles, and those are just going to bang around in the lumen until they come close enough to the enterocyte to cross the apical membrane by simple diffusion. Okay, so they cross the apical membrane by simple diffusion. Once inside the cell, they're going to associate with the smooth endoplasmic reticulum because that's where a ton of fat is. Okay, the smooth endoplasmic reticulum is going to reform them into triglycerides. Those triglycerides are going to get sent to the Golgi apparatus, which is going to package them with protein. So you're going to put a whole bunch of triglycerides together, some protein. It's going to be put into a vesicle, and then it's going to be released by exocytosis. It's too big to get into the bloodstream, enters the lacteal instead, and then goes into general circulation, not directly to the liver. Okay? Carbs and amino acids go directly to the liver. Fat gets dumped into the vena cava, right, in the form of chylomicrons. We'll stop there for today. Right, so we're going to start talking about some of the accessory digestive organs. So these aren't part of that tube that runs from your mouth to your anus, but they play an important role in the digestion of your food. So your pancreas is considered an accessory digestive organ because its exocrine portion secretes pancreatic juice. And the pancreatic juice has a whole bunch of digestive enzymes, right, that digest all of the major macronutrients. So the pancreas releases proteolytic enzymes. The pancreas releases amylase for carbohydrates, and the pancreas releases lipase for fats. Okay, and then the pancreas also releases a whole bunch of bicarbonate in order to neutralize the superacidic chyme that is entering the duodenum from your stomach. Okay, so your pancreas plays a major role in digestion. We've already talked about the pancreas because it also has an endocrine function. Okay, so the endocrine function has from the islets of Langerhans, and those are those cells that secrete insulin and glucagon. And what do insulin and glucagon do? Regulate blood sugar or blood glucose. Okay, insulin gets released due to high blood glucose levels and causes absorption of glucose into cells. So it lowers your blood glucose levels, and then glucagon is released due to low blood glucose levels, and it causes cells to release glucose into the bloodstream. Okay, but what we're going to talk about is that exocrine function of the pancreas today. Okay, so what we're missing here is the liver. The liver basically blots out the sun. Okay, so the liver is missing in this figure. And also the stomach so that we can see the pancreas. So the exocrine portion of the pancreas secretes its products into the pancreatic duct. Okay, remember, exocrine glands secrete their products to an external body surface, which would be the pancreatic duct. Endocrine glands secrete their products into the bloodstream. Okay, so the acinar cells are what secrete the enzymes, the digestive enzymes. The duct cells are what secrete the bicarbonate. And then that pancreatic juice enters into the duodenum, and the stomach empties into the duodenum very close by. Okay, so as soon as that superacidic chyme enters, it's going to be neutralized by that pancreatic juice. Okay, the gallbladder secretes in the exact same area as the pancreas. Okay, and the gallbladder is the storage organ for bile. The liver itself makes the bile. Okay, so the liver makes the bile, and then it gets stored in the gallbladder, and then the gallbladder will contract when you eat a fatty meal and secrete bile into the duodenum as well. And we'll talk about the gallbladder next. Okay, so your pancreas secretes enzymes that digest protein, digest carbohydrates, as well as lipids. Okay, so the proteolytic enzymes that break down peptide bonds are trypsinogen, chymotrypsinogen, and procarboxypeptidase. Okay, so your pancreas secretes all of the proteolytic enzymes in an inactive form. Okay, just like your stomach. And so that's so that they don't digest those cells anyway. Okay, so they get secreted in inactive form. Trypsinogen gets activated by enterokinase. Remember, enterokinase was one of those brush border enzyme on your enterocytes. Okay, so trypsinogen gets released into the duodenum, and then enterokinase converts it into trypsin, and then trypsin will activate chymotrypsinogen, okay, into chymotrypsin, and then trypsinogen will activate procarboxypeptidase into carboxypeptidase. Okay, and then they're going to finish digestion of all the protein. Where does protein digestion start, though? In the stomach. Okay. All three of these proteolytic enzymes work best at a neutral pH. Okay, pancreatic amylase, just like salivary amylase, is going to convert polysaccharides into those limit dextrins and into disaccharides. Okay, and then the disaccharides are going to be digested by those brush border disaccaridases on the enterocytes and then absorbed. Okay, and then pancreatic lipase is the main enzyme that digests fat. And because it's a protein, it stays in the watery matrix. So we have to emulsify the dietary fat so that lipase has a greater surface area to work on. Okay, and it breaks down those triglycerides into monoglycerides and fatty acids. You do secrete a lipase in your saliva. It's called linguinal lipase, but it does very little fat digestion. Fat really isn't digested until it hits your small intestine. Okay. Another accessory digestive organ is your liver. Okay? Your liver is the largest and most important metabolic organ in the body, and we're going to talk about it for only a couple slides. Okay? But you should all thank your liver. Right? It is your major biochemical factory. Okay? It receives all the nutrients absorbed from your small and large intestine on what's called first pass. Your liver gets first pass. So there is a portal system between your small and large intestine and your liver. Okay, and a portal system is just two capillary beds connected by a vein. And the vein that connects this portal system is the hepatic portal vein. So everything you absorb from your small intestine and large intestine goes to your liver first. And if it's toxic, your liver will metabolize it. Okay, that's one of its biochemical functions. Right? If you take in an excess amount of iron or carbohydrates or amino acids, your liver can also serve as a storage organ. Okay, and store it as glycogen if we're talking about carbohydrates. Okay, your liver is super active. So it's seeing what you just absorbed from your GI tract, but it needs oxygen, right, and nutrients itself constantly, and it's getting that from the hepatic artery. Okay, so it's getting its oxygenated blood from the hepatic artery, which branches off of the aorta. So you've got two blood sources for the liver. We're talking about it with the digestive system because it makes and secretes bile salts. Right? So that's its major role in digestion. Okay. Anything that gets absorbed across your stomach lining doesn't hit your liver first. And one of the things that gets absorbed across your stomach lining is ethanol. Right? Males produce an enzyme in their stomach lining, alcohol dehydrogenase, so they actually metabolize some of the ethanol as it crosses their stomach walls. Females don't, right? And that's why females will get drunker faster, especially on an empty stomach, right? So, right, the liver does a whole bunch of things. Some of them are not related to digestion. Okay, it processes the major categories of nutrients. So it processes lipids, proteins, and carbohydrates. Okay, it detoxifies or degrades body waste, so hormones that are no longer needed. Okay, any drugs you take in, as well as other foreign compounds, so food additives, etc. It makes most of your plasma proteins, except for your antibodies. What cells make your antibodies? Plasma cells, okay, which are B cells. It is a major storage organ. It stores glycogen, fat, iron, copper, and the fat-soluble vitamins, so A, D, E, and K. So that's why before we had multivitamins, liver was a really common food. People probably ate liver once a week, right? You guys probably don't eat a lot of liver because it's kind of gross, although maybe you do like liver. I don't want to judge, right? But I would much rather take a multivitamin than eat liver. Hey, your liver plays a role in activating vitamin D. And what does vitamin D do? The activated form of vitamin D plays a role in calcium balance, okay? Increases absorption of calcium from the GI tract, as well as reabsorption of calcium from the kidneys. Okay, your liver will remove bacteria and worn out red blood cells, especially if your spleen is damaged. Okay, you think of your liver as a big filter organ. It also excretes excess cholesterol, and it excretes bilirubin. And what's bilirubin a waste product from? Erythrocytes, so red blood cell breakdown, okay? And it's what gives bile its brown color. Okay, so here we're showing that portal system where you have the capillaries, right, that are in the small intestine as well as large intestine, and then they're connected to the liver via the hepatic portal vein, okay? And what this is showing is that the liver, when it makes bile, one of the major components of bile are bile salts. So we've got these bile salts here. Remember, they're the amphipathic molecules. They're made from cholesterol. So they have a hydrophobic portion and a hydrophilic portion. Gallbladder acts as a storage organ for bile. Okay? And when you eat a meal, especially a meal high in fat, the gallbladder will contract and release bile into the duodenum. And then those bile salts will get reabsorbed in the ileum unless you eat a lot of soluble fiber. Okay, and oats are really high in soluble fiber. So that's why your Cheerios or your oatmeal container get to put that heart-healthy heart, right, on their label, because soluble fiber irreversibly binds your bile salts so that you don't reabsorb them. Okay, and then you'll just remake them from more cholesterol. So it's a great way to get rid of excess cholesterol because you end up excreting that cholesterol. But it's only soluble fiber, okay? So oats are what irreversibly bind those bile salts so that they don't get reabsorbed in the ileum, okay? As well as any cholesterol that gets excreted. Okay, so the last organ of the GI tract we're going to talk about, we're going to go back to the tube and talk about the large intestine. Your large intestine is primarily a drying and storage organ. Okay, and it consists of your colon, your cecum, your appendix, and your rectum. Okay? It's getting anything from the small intestine that wasn't digested and absorbed. Okay? And what is not digested or absorbed in the small intestine? Ever. What? Fiber. Exactly. Okay? So to keep your large intestine happy, you're supposed to eat lots of fiber. Okay? Does fiber get digested in the large intestine? By us? No, by microbes that are hanging out in there. Okay, so when you eat fiber, you're basically feeding those microbes that are hanging out in your large intestine. Okay, anything that was excreted in the bile and didn't get reabsorbed will end up in the large intestine. Okay, and then there's often fluid that then gets absorbed in the large intestine. Okay, so one of the major roles of the colon is to extract more water. Okay, so that you don't produce watery feces, i.e. diarrhea, as well as salt from the colon contents. Okay, and what gets eliminated is your feces. Okay, so here we've got just the colon showing, or the large intestine showing. Okay, we've taken out the small intestine. So there is a sphincter between the small intestine, the ileum, and the ascending colon, cecum, and appendix area, and that's the ileocecal sphincter. Okay, it will open and allow contents from the ileum to enter the large intestine, which then is going to go up the ascending colon, across the transverse colon, down the descending colon. Anything that wasn't digested and absorbed is going to end up in the rectum and then be released to the external environment. And there are two sphincters holding in the feces, right? Your large intestine is colonized by beneficial bacteria. You are housing about 1 kilogram of bacteria in your large intestine. Okay, and I don't think this is correct at all. They're saying that there's between 500 and 1,000 different species present. It's probably a lot higher, right? And this is a super active area of research right now. You might have heard of the microbiome. Right? There's more genes in your microbiome than in your cells, okay? And they're proclaiming that the microbiome can, alter everything. Whether or not that's true, I'm not sure. But it is playing a major role in your health. So when you go on antibiotics and you kill off a bunch of the microbes in your GI tract, you're often not very happy for a week or two afterwards. Okay, so that's why oftentimes when you go on a course of antibiotics, they tell you to do probiotics as well. So have things with active cultures, things like yogurt, right, are very popular right now. And the reason why is because your bacteria help enhance your immunity in your intestine. Because remember, your gut is your largest immune organ. And those beneficial bacteria help outcompete the pathogenic ones. Okay, so by having beneficial bacteria, you're less likely to get pathogenic bacteria. Okay, they promote colonic motility. And there is a whole line of yogurt, that Activia yogurt, that has a specific species, the Bifidus regularis, right, bacteria that helps promote colonic motility. Right? That's the name of the bacteria. They discovered the new species and they named it Bifidus regularis so that you poop regularly, right? Okay, your bacteria help maintain mucosal integrity so that the lining doesn't get too leaky. Okay, that bacteria ferments the fiber. Okay, so your gut, your large intestine is anaerobic. And the cellulitic enzymes in those bacteria, right, help to digest that cellulose. And then the byproduct of that digestion process is volatile fatty acids. And these can cross the lining of your large intestine. Okay? So a lot of the crosstalk between the bacteria in your large intestine and inside of your body is via these volatile fatty acids. Okay? And different bacteria make a different profile of volatile fatty acids. Okay, your bacteria produce vitamin K for you. And that gets absorbed in the large intestine. Okay, and then those bacteria decrease colonic pH. They make it a little acidic, and that helps with mineral absorption. So you do absorb calcium, magnesium, and zinc in your large intestine. Okay, so there's lots of benefits to housing that kilogram of bacteria in your large intestine. So it's good to feed them properly. So eat lots of fiber. All right, any questions about the GI tract? Okay.